Thank you very much, and good afternoon. I'm going to jump in right away because inevitably I made too many slides, and I feel dearly about each one of them, so I just couldn't cut any more. But the goals of this lecture, by the end of this lecture, you should be able to list the advantages of molecular methods over conventional microbiology and virology. You should be able to describe how testing in public health events differs from testing for patient diagnosis. Listing the difference for several molecular techniques that we use quite a bit of in public health. And then one of the very important things is you should be able to define a clone in the way that we use it in terms of epidemics and outbreaks. In addition, by the end, you should be able to explain why all three of these pictures are put together like that. Okay, we're going to first have a pretest. We'll go through some definitions. You've never had an epidemiology lecture that didn't have something to say about John Snow. We'll talk about epidemiology and laboratory synergy. I'll review some of the molecular methods that we do use in public health. I'm going to give you 3 public health vignettes to show you how the molecular tests have changed the way that we approach some of these public health problems, and then the summary. First, though, we're going to start out with the pretest. And the question here is, what kind of scientist are you? I am going to show you for 1 second 4 images from the Ebola outbreak in Kitwe, Zaire in 1995. And it's just going to be 1 second, 1/1000, okay? Now, we're going to find out what kind of scientist you really are. If you looked at that and you zeroed in on this man and you said, how can I change this man's outcome? You are a physician. If you looked at it and you looked at these people first, you said, gosh, I wonder how many of these people are really infected, you're an epidemiologist. If you exclaimed, what an exquisite filovirus, you're a basic scientist, and I'm just going to ask you not to snore or laugh during my lecture over the course of the next hour. And if you said, is this Ebola Zaire virus the same as the Ebola Zaire virus that infected chimpanzees in Cote d'Ivoire in 1994, you're a molecular epidemiologist, and this is the world we're going to live in for the next 40 minutes, talking about how we show relatedness to solve public health problems. Let's start with a few definitions. There's only 2 that we use quite a bit of. One is transmission, and that is just what all of you know it as. It's the movement of an infectious agent from a person, animal, or in the environment to a person. And so we talk about fecal-oral transmission. We talk about aerosol transmission. And we document that transmission occurred when we can show that the same organism is present in 2 people, or the same organism is present in an animal and a person, or in the environment and a person, and then documenting that connection. So here, okay, use your pointer, you told me. Okay, come on, there it is. Okay, we're talking about the transmission of influenza and a whole host of other viral respiratory pathogens, Legionella, Pseudomonas. In the lower corner there, that animal with the nasal secretions, that's Rift Valley fever. The wrestlers in the middle, that's herpes gladiatorium. Next to them, that's a Culex mosquito, the vector for West Nile virus. And next to that is the Asian rat flea, the vector for plague. So the way that we know these is that we were able to demonstrate that the same organism that caused disease in a human being was also found in these other situations. The other concept that I want to define is a clone. A clone, the way that we use it, is a cell or group of cells or an organism that is descended from and genetically related to a single common ancestor. Bacterial colonies all start with one bacterium and they subdivide. Now, if I told you these imperial stormtroopers were a clone, or that Homer Simpson was a clone, or Dolly there was a clone, you'd all believe me because they all look alike. But if I told you that all 3 of these organisms in the lower right-hand corner, that is Salmonella paratyphi, collected in 3 different patients were clones, you'd say, you have to demonstrate that to me. And that's what we're going to be talking about again now for the next 45 minutes. As I said, no lecture in epidemiology or public health ever goes on without talking about John Snow. John Snow was actually trained as an anesthesiologist. He gave Queen Victoria anesthesia twice during labor. It was the first time that it had ever been used, that chloroform had ever been used in labor. But despite that, and he became a very rich man because he was the anesthesiologist to the Queen. Despite that, we know him as the father of epidemiology. In 2003, he was voted to be the greatest doctor of all time by Hospital Doctor magazine, which is a throwaway magazine in the UK. I will tell you that there was a great deal of ballot stuffing that was going on by the John Snow Society, and it was probably the only way that he could have beat Hippocrates out for the #1 position. He is best known for his 1854 cholera work in London, where he was able to show, using both rates and statistics, that a particular area, a particular point, what we know as the Broad Street Pump, was the source of cholera for a very, very large neighborhood, causing many fatalities. He did this using what I've come to term the LPS, the London Positioning System, instead of the GPS. And he has been absolutely critical in terms of showing that science-based public health decisions are possible. He worked very hard to identify points of intervention. And then one of the most important things, and things that we're still learning about today, 150 years later, is that it is important to measure the effectiveness of your intervention. A little about cholera pandemics at the time of John Snow. Cholera is a diarrheal disease due to Vibrio cholerae. We often will say that it was discovered by Robert Koch, but in fact Filippo Pacini probably discovered it in 1855. It's just that Robert Koch had better press. These outbreaks at the time were attributed to miasma, or bad air, and not germs or bacteria. The cholera pandemics had never come out of Asia until the 1800s, and during that time we had 3 pandemics. The one that we're going to be talking about is this third one from 1852 to 1869. Probably the most important thing to know about cholera is it has a very, very specific clinical presentation, unlike a lot of the other diarrheal diseases. London in 1854 was just awful. The River Thames was heavily contaminated. It was a rapidly growing city. They had a water delivery system that was absolutely necessary to move water from the Thames to a lot of the peripheral neighborhoods, and the River Thames was extremely contaminated. It was so contaminated that in 1852, Parliament ordered the water companies to stop their intake from the middle of the Thames in the middle of the city to upstream from London, so that they were pulling clean water instead of already contaminated water. One of these companies, Lambeth, complied very quickly, but the other company did not. They waited until the deadline. And this was actually possible then to look at what John Snow called a grand experiment. I'm not going to go into that one today, but it is actually a second cholera investigation in which he was able to show the impact of cholera on very similar communities with 2 different water sources. And another piece that was very, very important in terms of thinking about the development of epidemiology as a tool for public health. So let's start with the Broad Street cholera outbreak. This was the quote from John Snow in September 1854. And the key points here are highlighted in yellow. Within 250 yards of the spot where Cambridge Street joins Broad Street, there were upwards of 500 fatal attacks of cholera within 10 days. And I'll show you a graph of that here in just a bit. Now, if you had a report like that, you would be calling up the Department of Health, and we would have our environmental scientists down there sampling the water. We would have our laboratorians coming together and thinking about the best way that they were going to demonstrate what the organisms are, and we would have an epidemiologic investigation. But unfortunately, in John Snow's time, those things were not available. You have to remember that we didn't even know that cholera was caused by a bacteria. He had an epi-investigation. And these were his methods. John Snow did a retrospective death record review. He went down to the registrar and identified all the deaths that were due to diarrhea and cholera. He then mapped the location of each of the residences without satellites and without GPS. He presented those data to the Board of Guardians of St. James Parish and to some of England's staunchest miasma proponents, who decided to put an intervention in place. The myth and the fable is that John Snow snuck over to the pump in the middle of the night and pulled the handle off it like at 2 in the morning or something. But in fact, this Board of Guardians did reluctantly put in place the intervention. And then Snow put in place follow-up surveillance to measure the effectiveness of pulling that pump. Here is a recreation of his original map. The red dots show pumps that were distributed all over the entire neighborhood. The brown or beige dots here show deaths. And you can see that there is an increased density of deaths around that red dot that is there on Broad Street. That is a replica of the actual pump. And it actually does not stand in the place where it actually was in 1854. But what John Snow did is he mapped each one of these deaths, and he was able to show that the density, the case fatality ratio around this pump was much greater than any of these around any of these other pumps. And then he did a review of the first 83 cholera deaths, and he in fact showed that all but 6 of those had used water from the Broad Street pump. Another point that he had made is that there was a brewery in the middle of this area that was less than 100 feet away from the Broad Street pump, and despite its proximity to the pump, that there were no workers who became ill, and that is because they had their own water source and because they drank a lot more beer than they drank of water. Here is what we call an epidemic curve, and now I'm going to start introducing the concept of clonality. What an epidemic curve is the number of cases that occur, in this case, over time between August and September. And what you can see is that there were about 500 total deaths from that time. And I have something marked there called the index case. That is when baby Francis dies. Baby Francis is a 4-month-old baby who dies of choleric diarrhea that had gone on for about 4 days. During that time, she had frequent bowel movements because cholera just has an incredibly profuse output of diarrheal stool. And mom was washing out her diapers very, very close or to a cesspool that leaked into the well of the Broad Street pump. So this goes on, all these deaths are going on over about a 2-week time period, and then finally the pump handle is removed on September the 8th. Now, one of the questions that we would ask if we were the editors reviewing this for a journal is we would say, well, how do we know that the same organism that caused cholera in the index case is the same cholera organism that is causing the deaths that occurred, say, on September the 6th? How do we know that they didn't have another source of exposure? Why are you attributing it to the Broad Street pump? And those are the questions that we had today. Those are questions that were not even possible at the time of John Snow. I will tell you, and this is somewhat heresy, that John Snow was one lucky guy. He was very lucky that he didn't over-anesthetize Queen Victoria to begin with. Not only would he have changed the course of human history and potentially the history of World War I, which occurred 60 years later, but more importantly, he would have changed the whole epidemiology theory and concepts. We would have a completely different way of approaching these things. But he was very lucky because cholera is clinically distinctive. There are very, very few enteric pathogens that have a similar clinical presentation. In addition, during this particular outbreak, transmission was extremely efficient. You have to consider that the drinking water from the Broad Street pump must have been a huge risk factor. I mean, there are hundreds and hundreds and hundreds of deaths. We haven't even talked about the illnesses in people that have recovered, so it had a very, very high attack rate. In fact, in some of those blocks that Snow mapped, there was a 20% mortality just due to cholera alone. He was also very lucky because although the officials at the St. James Health Parish were not convinced that it was the water, they did remove the pump, and it was a very, very important step. But it does underscore some things for us today when we start talking about some of the testing that we are going to use in public health. And that is that we really need to be more specific about our diagnosis. That even though Broad Street was a very, very successful investigation, it did use clinical case definitions. And today there are still some diseases that are pretty much pathognomonic in terms of their clinical presentation. Cholera, plague, TB, yellow fever, the childhood exanthems, polio, smallpox. When you see one, you know one. You don't necessarily need a test to do those things. But most of the diseases that we deal with as epidemiologists, most of the diseases we deal with in public health, in fact, do not have a unique clinical presentation. And some of these syndromes do not have a single causative organism. So think about it. Why do we at the public health agencies always talk about influenza-like illness? Well, it's someone with a fever, it's someone with a cough, it's someone with myalgia and a headache. But there's 16 different viruses that have been documented to have these influenza-like illnesses. And there's other diseases like that. Typhoid, very, very nonspecific clinical presentation. When bacterial meningitis occurs, there's 3 or 4 agents that we should think of automatically. You don't know which one that is. Diarrheal disease is in that same category. So what we come away with from Broad Street is that we really need to specifically identify the causative organism if we are going to specify the mechanism of transmission. Nonetheless, Broad Street was significant. And it's important to epidemiology, to laboratory medicine, and to public health. Laboratory medicine because it is the start. We started saying we need a little bit more if we're going to start approaching other diseases like we've approached cholera. It was very important because it showed that germs and contamination cause infectious disease. And although it was not generally accepted for about another decade in London, but the Broad Street events really did win over many of the miasmaists that were present in London. And as I said earlier, it is a very important step because it shows that the scientific process can be applied in public health decisions, and that was revolutionary at the time. So we come away from Broad Street looking at a disease control cycle. First, we identify a cluster. What is a cluster? They are people with the same syndrome, clinical syndrome, during the same time period in the same place. And you have to ask yourself now, as laboratorians and molecular epidemiologists, well, what is missing? Is it caused by the same organism? And that is something that he could not answer then, and over the course of the next 110 years since Broad Street, that is where there's been a great deal of emphasis. When the rest of this disease control cycle is that we should identify risk factors for transmission, we should design interventions to stop those transmission, we should measure the effectiveness of that intervention, and then we need to revise our control methods as needed. All of this from Broad Street. Let's talk a little bit about epidemiology and laboratory synergy. From 1854, since the Broad Street pump events, to about 1970, we needed to move beyond clinical case definition. Why do I say 1970? 1970 was the first isolation of restriction enzymes, and that revolutionizes what we know in terms of molecular testing in public health. The whole reason that we needed to move beyond clinical case definition is, as I mentioned, that different pathogens with different transmission mechanisms can cause the same syndrome. So we need to identify the specific organism. So what laboratory does over the next 116 years is that they are moving to confirm that cases are due to a specific agent, and they do it through microscopy, through culturing these organisms in animals, culturing these organisms on artificial media, the use of anti-sera to serogroup and serotype. We still do it today in a lot of situations. Viral cell culture in the 1920s and the 1930s. Serology testing also in the 30s and the 40s. And is still getting new technology applied to it today. And then antibiotic sensitivity profiles. So you can see that people are working harder and harder to show what is the organism? Are they the same organisms? Are they clonal organisms? Despite the progress in the laboratory clinicians and epidemiologists still lived in different worlds. There was not a great deal of cooperation. And I think, I don't know how many of you have read The Great Influenza, but that first 100 pages of that book just talks about how laboratorians really didn't see a lot of worth in terms of dealing with trained physicians or epidemiologists. I mean, they were moving along on their own pace. What had to happen was somebody had to come along and say, you know what, the laboratory and the epidemiologists really do have to work together. And this happens in about the middle of the 20th century. By 1949, it was generally accepted that the Center for Disease Control efforts had become quite antiquated. CDC had begun primarily as a wartime agency to stop malaria transmission in the South. and to control malaria. So as a result, they had a lot of entomologists, they had a lot of vector control people. They did not have many laboratorians. They didn't have many epidemiologists, and there were probably only 2 or 3 physicians in the whole place. Dr. Mountain is a physician who comes to CDC in 1949, and he realizes it's time to bring in people who know laboratory. It's time to bring in epidemiologists. And it was at this time that Alexander Langmuir, probably one of our foremost epidemiologists in this country, was hired in the CDC. 6 years later, they get the call. This is the event. This is the thing that's going to bring together and show epidemiologists how much they need laboratorians and how much laboratorians need epidemiologists. In April of 1955 in California, there were 5 children that developed polio after being vaccinated with the polio vaccine, the inactivated polio vaccine. The CDC epidemiologists went in, they applied Broad Street pump principles, were able to identify a suspect lot of Cutter inactivated polio vaccine. The lab took those lots and tested them, and they showed that in that particular lot that there was inadequate dwell time with the formaldehyde, and that in fact live virus was still present in that lot. And what that did, not only did it bring the lab and epidemiologists together, it restored confidence of the public in polio vaccine, which was very, very necessary at the time. But this is really one of the first great examples of cooperation between laboratory and epidemiology. So, why do epidemiologists need laboratorians? I think I've made the point that multiple etiologic agents are present for the same syndrome, and we need to know about the specific etiologic agent when we're talking about intervention. Why do laboratorians need epidemiologists? Well, I hate to say that laboratorians are using us, but what they need us for is really to provide the proper samples for testing. You know, unless you are out there investigating each one of these cases, you don't know that you're necessarily bringing the proper samples back. You're not necessarily receiving the proper samples in your laboratory. So what epidemiologists do is they help laboratorians who are involved in public health focus lab testing. Who's truly exposed, who truly has illness, who truly should be tested, and then of course lab validation. I'm not going to go into the advantages or disadvantages here, except to point out one thing. We are moving very rapidly towards a time where people are saying, why don't we use molecular testing for everything in epidemiology? I will tell you that there are many things about the intact organism, about being able to culture an organism, about being able to have lots of organism, and that is very, very important in terms of epidemiology, especially when we're talking about the study of the pathogenic factors that are not proteins or genetically coded. And so it is a very, very important thing to get isolates. But in terms of a lot of the other things that we do in terms of showing the clonality, showing the relatedness of organisms in different people, that very often these things are being replaced by molecular methods. Let me go through a quick review of some of the methods that we use to support public health decisions. There's really 3 broad categories. There's fingerprinting techniques, and I will talk about a few of these here in a bit. And fingerprinting techniques can use either culture product or nucleic acid amplified product. The ones that we use a lot in public health are pulse field gel electrophoresis, PFGE. We don't use multi-locus enzyme electrophoresis anymore. Restriction fragment length polymorphism. I think a lot of people have thought that it had died a death, but in fact, you will still see new methods in which they're using RIFLIPs. MELVA, Miru, spoligotyping. I will not talk about Miru or spoligotyping, but I will talk a little more about MELVA, but there's new ones coming online all the time. So there are those categories where we can actually do functional characterization of the organism. Antibiotic resistance patterns. When we say that 2 organisms had the same antibiotic resistance pattern, and therefore they must be the same organism, what we're saying is that we've characterized them functionally, and they sure look and act alike. So more likely than not, they are a clone of each other. Doesn't always hold, but it's sometimes what we use. Microarray is being increasingly used in terms of the functional characterization. But the things that are really going to change the ballgame right now are the sequence-based methods that we're using. That's multi-locus sequence typing, MLST, pyrosequencing, and single nucleotide polymorphism. There are others out there as you move through, but these are the big ones that we tend to see occurring again and again in public health. There are many advantages to molecular tools: rapid identification of the organism, rapid identification of a toxin, rapid identification of antibiotic resistance. The one that we use an awful lot as molecular epidemiologists are the clonality. The molecular tools give you the ability to do multiplex testing, which is very difficult to do when we're talking about more conventional microbiology or virology. We can identify co-infections. We can identify pathogens that cannot be cultured. And we can identify new pathogens. So I'm going to talk a little more about these four particular methods. Okay, first, RIFLIP and PFGE. RIFLIP is a gel electrophoresis to separate DNA fragments created by restriction enzymes. PFGE is a gel electrophoresis to separate DNA fragments created by restriction enzymes. What's wrong with that picture? In fact, they are really basically the same technique. But these are the differences. And one of the big differences is in terms of the DNA fragments that result from the use of the restriction enzyme. In RIFLIPs, they usually find a motif of 4 bases, and that's where the restriction cut occurs. Because it is only 4 bases, that means there are a lot more targets in that nucleic acid strand, and you're going to get a lot more cuts. The more cuts you get, the more smaller DNA fragments you get. PFGE generally uses restriction enzymes that look for a motif that are 6 bases to 8 bases long. As a result, there's fewer targets in a genome, and you get fewer larger DNA fragments. That's probably the most important difference between the two. In addition, though, there's a little bit of a different electrophoresis method. In RIFLIP, it is a continuous use of an electric field, and they let those fragments just kind of drift towards their respective poles. In PFGE, it's different. Because these fragments are much larger, you actually have to move them around through using a sieve-like action through the gel. And so what a pulse field gel electrophoresis machine does is it reverses the polarity, and it changes the shape, it changes the orientation of the nucleic acid fragments. The most important thing to me as an epidemiologist is the interpretation. In terms of a RIFLIP, and I'll show you an example of that, very often you will need to use DNA hybridization to create identifiable bands. And with PFGE, there's very often a easily identified band pattern. So here's a schematic. On the left is the example of a RIFLIP. On the right is a pulse field gel electrophoresis. The golden triangles are a restriction enzyme cutting site. As I mentioned, you will find a lot more cutting sites in when you're using a 4-base motif cutting enzyme than you would in a PFG where you're using a 6 or 8 base motif. Okay, so I've demonstrated there are a lot more cut sites in the RIFLIP. And so this is the resulting band pattern. Now, when I look at that RIFLIP one, that would be very difficult for me to read just because there are so very many bands. Okay, there are some subtle differences, and in fact, B and C are identical to each other. But just looking at that quickly, and depending on a little smear in your gel, you may or may not be able to truly appreciate that. In contrast, the pulse field gels truly do create widely separated bands. And this is what I meant when I said that you can identify PFG very often by the banding pattern. But for a RIFLIP, you often will have to use a hybrid. You will have to hybridize it with another DNA molecule for particular segments. And now you can see that B and C truly are identical to each other. An example of that is the insertion sequence 1610 that you see when people are doing the RIFLIP for TB. That is a reporter that they have hybridized on there to show where that IS 1610 shows up in all of these fragments. I'm going to show you a picture On the top is an example of a riflip, and you can see how many bands there are. There is no distinctive band pattern. This is one that has not yet been hybridized with a reporter yet. On the bottom is a PFGE, and you can see how the bands are a little more distinct. Even when you use PFGE, though, in very large genomes, such as Mycobacterium fortuitum, you can see that there are lots and lots of bands, and sometimes it can be very, very difficult to interpret. And then, again, this is just an example of showing some of the banding patterns. This one is looking at some penicillin-resistant strep pneumoniae. The second technique that is being used more and more is the multi-locus sequence typing. MLST is really the molecular analog of the multi-locus enzyme electrophoresis, which uses banding. And instead, MLST, rather than using bands, is actually sequencing these genes, these housekeeping genes that had been spread in electrophoresis for MLEE. Instead, we're going to go ahead and sequence those, and I'll show you an example of that. Here is an example. On the left is a schematic of the multi-locus enzyme electrophoresis. What you do, and this is a great simplification. I looked all over for good pictures of multi-locus enzyme electrophoresis, and there are not many. I had to go out and get a cyprinid fish, which is a stomachic fish of Southeast Asia. I just found that out to show you an example, because usually they're not drawn. They will usually show you with the tree that results from the MLEE. I've had the fortune of sitting with two great MLEE folks, Francisco Ayala at UC Davis, and then Mike Reeves at CDC, and they look at these things and they're seeing things that I just don't see. There's a lot of art to reading some of these. So I've made a schematic on the left for you. Because what you're doing is really taking these housekeeping genes. And these housekeeping genes, because these are very, very highly conserved. They don't change very quickly. And you're really doing what we call isoelectric focusing. You're finding the isoelectric point. You're letting the overall charge of these enzymes move along this electrophoretic gradient, and they're finding their characteristic place. And if there is a significant change in one of those proteins, meaning that a positive amino acid goes to a neutral amino acid or to a negative amino acid, then that isoelectric point is going to change, and you're therefore going to have a different fingerprint. As you can imagine, though, as you all remember from your basic genetics, you can have a lot of wobble in the third base of the codon and still end up with the same amino acid. You can end up with a two base change in a codon, and you're going to end up possibly with one positive amino acid just replacing another positive amino acid. So no net change, and it's still going to come out to the same place in terms of the pattern. So even then, this is a great advance when it came along, and it was used extensively in public health in about the mid-80s, but it is simply not used anymore. Because instead, we're starting to use the MLST. And here's an example of an MLST. What they have done is they've looked at 7 genes, and they're looking at the internal fragments, usually about 400 to 500 base pairs long. These are the same 7 genes that people were using, or these are the genes for the same 7 enzymes that people were using within MLEE. And they were looking for some of the base changes. And then they were assigning a number to that. So now you can see that we can actually assign a clonal type to each one of these strains using simply a 7-integer number. And a great advance. The greatest advance, though, is that now you can be looking at base changes, not amino acid changes. So you could really appreciate much more subtle changes over the course of time. Multilocus variable copy numbers of tandem repeats, or MALVA. Some genes have multiple copies that sit in tandem within a genome. And within a particular species, there could be a large number of copies, a large range of copies, even among highly related bacterial strains. What MALVA does is it actually counts these number of tandem repeats, and then it compares the number of tandem repeats between different bacterial strains. And here's a schematic. We have a strain A in which we have 5 tandem repeats of the blue gene. We have 2 tandem repeats of the green gene, 5 tandem repeats of the yellow gene, and then 3 of the red. Again, you see what we can do is we can give it an integer designation, which makes it very, very easy in terms of some of the numeric software that is used today, rather than the banding, which is a much more difficult numeric. You can see that strain B here is different in that it has fewer tandem repeats than in the blue gene, more tandem repeats in the green gene, the same tandem repeats in the yellow gene, and more tandem repeats in the red. I'm going to go through a few public health vignettes over the next 5 minutes just to show you how these things are useful to us. Starting in May of 2010, CDC noticed a 4-fold rise in cases due to Salmonella enteritidis, a non-typhoidal serotype of Salmonella. In May or June of 2010, Washington State also had a doubling of reported cases of Salmonella enteritidis. In August, on the national level, shell eggs were implicated and a recall of eggs began. And we got a call from a reporter at the Seattle Times and called and says, asked us if Washington is part of this national outbreak and are we getting ready to recall eggs? So the question I have is, can molecular testing help answer these questions? By the way, of the 3 vignettes, 2 are true, 1 is a modified true, and I've designated them as such. Well, the way we best answer this question is using PulseNet. PulseNet is just an unbelievable change in the way that we approach outbreaks, and it is a really very remarkable laboratory epi model. Since 1996, we've had 63 state local public health laboratories and 12 federal laboratories that form a national lab-based network, surveillance network, that all use the same methods. They use the same method of PFGE. They use the same software to analyze the banding patterns. They look at 6 foodborne illnesses: Salmonella, Listeria, Shigella, Vibrio cholerae, Shiga toxin producing or E. coli, and Campylobacter. And it's based on the premise that just knowing the species and knowing the serovar is not enough to put together clusters. So, as I said, they use the molecular methods to identify the clusters first. We have just completely reversed what we had been developing for the previous 150 years, but it has revolutionized the way that we approach foodborne outbreaks. As I said, these are standardized methods for performing them that are done in all 75 of these laboratories, and standardized software. So here's the way that we applied it. This is a federal national map, or a national epi curve, showing the number of cases that have a particular PFGE pattern A that was reported by PulseNet in 2010. And you can see the 4-fold increase that I mentioned. Here's an epi curve for Washington State. You can see that tall peak right there. That is October of 2008. And I'm going to draw your attention to the far right end here, marked outbreak period. That's the outbreak period that where we're asking this question now. And you can see that yes, we did have an increase in Salmonella enteritidis cases, but in fact, we did not see the concomitant change in the PFGE pattern A that we saw on a national level. So, did PFG help us make a public health decision? Well, we know that SE serotype cases are common. In Washington, we get about 10 of such cases a month. In fact, the PFG pattern A is a very common clone within Washington. We get about 5 of those a month. And we did experience the increase in Salmonella enteritidis cases, but we did not see a specific increase in that particular pattern. As a result, we said, no, Washington is not part of this national outbreak. No, no recall is going to be done. And in fact, subsequently, we never did identify any links to any of the national cases. So, in fact, the PFG did help us. Let's go into a second public health vignette. We're going to go back in time here to Oregon, 1993 to 96. They had over 750 cases of meningococcal meningitis. Over 70% of these were due to serogroup B isolates. Using the older technique, multi-locus enzyme electrophoresis, we knew that these B isolates, many of them were clonal. It was what's called ET5. It was a clone that had moved up probably from Brazil into the Northwest and caused lots and lots of disease. In 1994, 10 days after there was a Group B meningitis case in a high school, a second case occurred in another student, and it was a Group C. As a result, nobody believed that it was a cluster, and so antibiotics were given to the close contacts of the cases, but broader antibiotic prophylaxis for the school, believing that everybody is now colonized with this clone or this organism, did not occur because these are different organisms, right? And vaccine was not considered for many reasons. Over the course of the next 10 months in Oregon, this happened on 4 more occasions. You got to ask yourself, what is up with that? And again, is there a role for molecular testing? And there was. And in fact, the story is well told in a 1998 article that's on the top where Marion Diermeyer and David Fleming's group, David Fleming is now the health officer for the county that you're sitting in right now, put together a great story talking about this outbreak. And Dr. Swartley in Dr. Stevens' laboratory in Atlanta, we were able to put together the story that this is in fact a capsular switch. And here's a table, and I apologize, I was not able to get a better photo, but if you squint your eyes really hard, you can see that of these 5 isolates that we're showing in this particular table, they all are the same serotype, they're all the same serosubtype, they're all the same immunotype, they all have the same MLEE pattern, 301. ET301 is a subset of the ET5 complex. And they all have the same PFGE pattern, but they have different capsules and therefore are put into different serogroups. The top 2 are a B and the bottom 3 are a C. So the come away story here is that in vivo we were able to show that even though these are clonal organisms, that in fact they're presenting with multiple serogroups, which is based on the polysaccharide capsule. And in fact, there was a replacement just in that particular gene that it was producing a C coat instead of a B coat. More recently, we've been able to use MLE's replacement, the multi-locus sequence typing, also for meningitis in Egypt and Sudan. And what we did is we randomly chose 86 isolates, CSF isolates of N. meningitis from Egypt and Sudan that had been collected in 98 through 2006, and they all underwent MLST. In those 86, we identified 19 different sequence types. Not surprising given that MLST is more sensitive to genomic changes than was MLEE. Within the group Bs, there were 13 sequence types that were circulating during this period, and within the group As, 6. But the interesting thing is that of the sequence type 2174, 8 of those were a group B, 4 were group A, and 2 were non-typable. Those occurrences in Oregon were not isolated events. In fact, you can demonstrate this all over the world, that within any particular sequence type, clones of each other, they can often have different polysaccharide coats. Now, ST2174 had first been reported in the United Kingdom in 1975, but almost disappeared from the rest of the world, and now it's almost exclusively reported to Egypt and Sudan. Why that occurs, still a subject of study, and underscores the importance of why we need to know where particular clones are. The virulence and the antibiotic resistance in these other organisms that we looked at did vary greatly by sequence type, not by serogroup. And we're preparing this piece right now for publication. But what this does is it undermines a lot of the traditional thoughts that we have with regards to the approach to meningitis, saying, well, if they're different serogroups, well, then there cannot be a clustered outbreak. So MLST was very, very valuable in this piece. We now have a website where MLST is broadly used. I have circled Dominique Cogant's name here, a Scandinavian researcher who did a lot of MLEE and is now really pushing for the use of MLST for more of these clonal investigations. Third vignette. 1996, over 150 cases of primary and secondary syphilis appear in high school students. There's a public health campaign to screen and treat the patients and widely distribute free condoms. The number of the cases greatly drops, but in fact, still 4 to 6 cases are occurring per month in this high school among white girls from affluent families. County supervisors are being called, of course, on a regular basis, and now they're calling you, and they want to know why we still have disease circulating in this somewhat unique and somewhat different group. And so the question we should all ask ourselves is, well, can molecular testing help us answer that question? By the way, this is close to being a true story, and I'll tell you how I modified it at the end. Syphilis is very difficult to investigate. The long incubation periods, the long latency periods, often early clinical changes are inapparent. There are almost no outer membrane proteins on treponema that make it very, very difficult to examine using immunofluorescence or any of the immunohistochemistry techniques. And T. pallidum still can't be grown on artificial medium. It still has to be passed in rabbits. And as a result, we do have limited genomic and enzymatic material available. But starting in 1988 and all the way up through last year, when a group here at the University of Washington has further added, we started using a lot more PCR. In 1998, Alan Pillay and our group, I was part of that group at that time, started using PCR for molecular subtyping of treponema. And then we started applying it in a lot of social networks. And I'll tell you a little more about what Sheila Lukart and Christine Mara have done here. With regards to molecular subtyping of T. pallidum, the first system was available in 1998. Pillay found that there were 2 genes that had significant variability, an acid repeat protein, and what he did is he applied single locus variable copy number tandem repeat analysis, or SILVA in that case, and then he did RIFLIPS for T. pallidum repeat gene. More recently, in November of last year, a University of Washington group said, you know what, we got to be moving to and adding sequence typing to that. Because one of the problems was the CDC technique identified a lot of subtypes, but there was one huge predominant subtype. And it was the most predominant subtype, so people really wanted to break that down into further categories. And what Christine Mara and Sheila did is that they were able to add something. And what they added is a single locus sequence of a particular open reading frame or a particular hypothetical gene. So what we end up with is an extremely complex and mixed system in which SILVA, RIFLIP, and SLST, not MLST, but SLST is being applied. This is the result of all of that. We have a CDC subtype on the left that shows the APR type. Okay, that was the variable copy number tandem repeat. The TPR type, which is the product of a RIFLIP, and then the sequence type that was added. So we now have this capacity, and this is a brand new technique. That has not been, well, actually, Sheila's paper does talk a lot about it in terms of the epidemiologic investigation. So, do we have a TrepNet coming down the line to parallel PulseNet? This is actually the story of the lost children of Rockdale County. It is a true story. The outbreak did occur in 1996, but there was no subtyping available at the time. There was no subtyping that was generally used until 2000 when we used it in the Carolinas and in Arizona. But had TrepNet been in place, it would have recognized very early on that there was a clonal outbreak in this particular county, and that transmission was primarily due to syphilis moving out of Atlanta into Rockdale County and Conyer High School specifically. So in summary, molecular methods do allow us to define clones. Investigating clonal clusters decreases the resources that epidemiologists need to do for case finding. For foodborne pathogens, PulseNet has allowed specific definition of a cluster based on PFGE. We're developing similar subtyping systems for TB, which I did not talk about today, for syphilis, for meningococcus, and a lot of other pathogens. And a lot of that, this is even going to come further because people will start applying pyrosequencers to perform their MLST as opposed to PCR. And then finally, using molecular lab results allows public health workers to move more quickly to intervention because we're able to rapidly identify the clusters, rapidly identify the transmission mechanisms, and put in place intervention. All right, so with that, these are the keys. This is one of your goals. You could see that the upper picture is Watson and Crick, the discoverers of DNA, and that's where we are now. That's where our molecular methods have gone. The middle picture is actually from a children's book called John Snow and the Broad Street Pump on the Trail of an Epidemic. And on the bottom is our future, and that's a pyrosequencer from Company Q, QIAGEN. I don't have any financial interest in QIAGEN, but I did cover their name nonetheless. But this is really where we're going to be going over the next couple of years. And with that, I'll take any questions that you have. Thank you very much. Yes, ma'am.
Question. I was curious about the meninge outbreak that you described, and so I always kind of think of the vaccine as being pretty reasonable for ACY and W138 or whatever it is. Is it your sense that now then when we see B, or is it actually, did it start off as B, or do you think there's some sort of selective pressure occurring in the host because the vaccine is targeting those other serotypes? I wasn't aware of such prevalent switching between the serotypes.
Well, the question is about whether the capsular switch could occur in situations, where there is some sort of selection pressure by vaccination. Excuse me. At that time, it was not. It was a B outbreak that had come up when that same disease, the ET5 disease, had occurred in Cuba, Oklahoma, in Brazil. It was a B, and then it got here. So vaccination never really came into play, and it is still unclear as to why a switch was made. Whether there was a co-infection with non-pathogenic Neisseria that also can code for a C polysaccharide, and that there was a sharing, and so there was just recombination events that have occurred. We don't know that. That is one of the things that we speculated on. Since that paper that we put together, it's been described in many, many places. It's been described B's to C's, C's to B's. And obviously our work in Egypt and Sudan, we're talking about A's are also involved in there. So it is a thought that there can be some vaccination pressure to change these capsules. And it's something that, in fact, our folks at the Department of Health and our lab are working with folks at the CDC to watch that question closely. Any other questions? Well, great. Thank you very much.